Sensory receptors. In this session, we will discuss sensory receptors. When we see sensory receptors, it is a sensory nerve endings that receives information and conducts a process of generating nerve impulses to be transmitted to the brain for interpretation and perception. The sensory receptors are vary in classifications but generally initiate the same process of registering stimuli and creating nerve signals. That is their basic function. When we see the specialized nerve endings or specialized cells, these act as specialized nerve endings or specialized cells. These are more sensitive to specific type of stimulus. These are able to convert stimulus energy into electrical energy, which is called as receptor potential. This can generate action potential in the nerve connected. When we see these sensory receptors, these detect changes in external or internal environment. That means these are detectable change is their stimulus. Who has the ability to detect change and how it is passed on to the CNS, how it is the process of transduction and specific type of energy is converted into electrical. When we see these different questions, there is seen the specific type of energy to receptor which alters the permeability of the cell membrane, which creates current flow, which causes changes at membrane potential, and following the changes in membrane potential creates generated potential. This is the excellent simple graphical presentation that explains us what is the basic mechanism of sensory receptors. When we see the different sensory organs, the sensory receptors with specialized non-neural cells like taste buds, this forms sensory organs. The structure, there are three types of receptors: bare nerve ending, capsulated nerve ending, and sensory organs. Now, bare nerve ending, these are specialized part of neuron. Capsulated nerve ending, these are connective tissue lamellae. While the sensory organ, these are specialized cells located at the end of afferent nerves. When you see this mechanism of receptor function, how this exactly act? It's very simple. There is an application of stimulus to the receptor. There is a change in the permeability membrane which creates graded depolarization, hyperpolarization, generate receptor potential, and which causes creates local current flow, that is electronic current flow, that is action potential. This flowchart explains us the basic understanding of the mechanism of receptor formation and which local currents or creation of local currents that creates action potential at the first node of Ranvier. That means stronger the stimulus, greater GP, and greater GP, that means higher frequency of AP in nerve ending. That means higher frequency of action potential in nerve fiber. That means when we see this GP is more stronger, greater the GP, higher frequency of AP, that means action potential in nerve fiber. Now, this figure shows us different types of cutaneous receptors like Merkel's disc, hair end organs, Meissner's corpuscles, Cross's bulb, free nerve ending, Fascinian's corpuscles, Ruffini's end organs. When we see this classification of sensory receptors, it's depend first group is depending on source of stimulus, second group is depending upon the type of stimulus, and third classification is basis of anatomical classification. These are the basic three types or groups of classification. First, depending on the source of stimulus, second depending upon the type of stimulus, and third is anatomical classifications.